Hello class, this is Adina with my brain dump on cytology, the study of cells. Specifically, we're going to be looking at human cells, including the cuboidal cell, the squamous cell, and your columnar cells. So your cuboidal cells are cube shapes like this, tends to be rounded edges. Your squamous cells are squashed, and columnar cells are column shaped. We're going to be using the cuboidal cell to demonstrate the various organelles and structures within the cell. Starting with your cell membrane. So our cell membrane is pretty special because it is made up of a phospholipid bilayer. What that means is you're going to have something with a phosphate head and a fatty acid tail, and two of them with the tails facing each other. The phosphate head is going to be hydrophilic and the fatty acid tails are hydrophobic. What makes these special is that they allow us to have a semi-permeable membrane. That semi-permeable membrane is going to allow some things to pass easily and other things not to pass so easily. Specifically, the things that pass pretty easily are going to be your lipids, your fats that are, so your vitamins that are phosphates, your neutral molecules, and your very small molecules will all pass through that bilayer fairly easily. The things that will not pass through very easily are going to be your large molecules or anything with a charge. Moving on from that, I am going to start with the nucleus in the cell. Put it in the center. It's not always perfectly centered, but I'm going to be doing that. So our nucleus is going to have a nuclear envelope. That nuclear envelope is also going to have that phospholipid bilayer. That phospholipid bilayer also has some cholesterol in it to help it hold its shape. Within that cell, you're going to have your nucleolus. And that nucleolus is where your ribosomes are synthesized. And you're going to have your DNA in your nucleus. Your nuclear envelope is going to have channels in it. The channel that we care about right now is the one that is going to lead to your rough endoplasmic reticulum. It's going to go like this, back and forth like this. And what makes it rough is going to be your ribosomes. These ribosomes are pretty special because they are all about reading genetic code and then synthesizing your peptides and your proteins, which are essential to life. But continuing on from that, rough endoplasmic reticulum is going to be your smooth endoplasmic reticulum, which is going to be slightly different shapes, kind of like that. Your smooth endoplasmic reticulum is not going to have the same thing, RNA on it, excuse me, I'm fighting a migraine. It is all about cleaning and detoxifying things coming off of that smooth ER is going to be your peroxisomes, and that's going to be using hydrogen peroxide to break things down. Moving on from that is we're going to be going to your Golgi complex, and that's kind of shaped like dinner plates like that there. Your Golgi complex is pretty cool. It is used to help transport things. It finishes also the synthesizing various proteins and carbohydrates. Your Golgi complex is going to also have these Golgi vesicles, and some of these Golgi vesicles are specialized and become your lysosomes. Your lysosomes is all about lacing things, and it is filled with digestive enzymes which can break things down. It can do things like break down sugars and release them into the blood. It also helps clean your liver. Lysosomes are very important and special. Moving on from that is, let's look at our mitochondria, our very special mitochondria. which, as everyone knows, is the powerhouse of the cell. But what does that mean? It means that it produces your ATP. So you're going to have an inner and outer membrane. The inner membrane is the squirrely one there, and it's going to be filled with these various caverns, which are those little hills there called cristae. Inside there is going to be your matrix. So it's synthesizing your adenosine triphosphate, which is used a lot in transporting, active transport. So things can go through that semi-permeable membrane and is just considered energy. Also, if you remember from various science classes, if you took them, if not, no judgment, it is where your Krebs cycle is taking place and your electron transport chain and a whole bunch of other things that some of us had to spend a lot of hours memorizing at one point in our life. Moving on from your, also it's going to have some ribosomes in it too. Let's use that same green color. Perfect. And this is special mitochondrial RNA. Another special thing about your mitochondria is it has its own DNA too that is passed down from your mother. So that's how you can follow like just a mother line, which is kind of cool. Going on from that, besides just some RNA floating around, is your centrioles. And your centrioles are going to be kind of shaped like this. And they're going to be perpendicular and always come in two. An easy way to look at them from a cross section, though, is they're going to be composed of microtubules, always in sets of 3. So then can attach there, and another 3 there to go all the way around. So you can kind of see them from a cross section in a microscope that way, which makes them really easy to identify. I think that is my brain dump on cells. Hope this is helpful to someone. It was for me. Thanks. Bye.